Title of our talk today, because I've dragged Tom in, because I am a supramolecular chemist and sometimes biologist, but I can barely turn on a computer, as we just realised by me trying to share my screen. This was not terribly laboured. So I wanted to bring Tom, who's an MRes student currently under the supervision of Jeremy Frey, who's actually opened my eyes to the world of artificial intelligence, machine learning, and data curation, because I have no idea what random trees and forests are. By that explanation, that's as far as I can go. So first of all, what I was going to do was tell you a little bit about what kind of scientist I am and what problems I have, and then I'm going to let Tom do the actual academic bit in the end, because he's far more qualified to do so than me. Right? So one is supramolecular chemistry. Supramolecular chemistry is the study of non-covalent interactions between molecules. It's a bit like this molecular Velcro here. So two molecules come together, they get stuck for a little bit, and what I do basically is study the strength of those Velcro-type interactions between these molecules. I am basically a baby biologist, although a biologist would say I don't really do biology because it's not really that complicated and my molecules are far too small. Okay. So back in 2015, I was lucky enough, 2020, I was lucky enough to be awarded a UKRI Future Leaders Fellowship for the development of this class of molecules shown here. These are supramolecular self-associating amphiphiles. They're very small, but what they have are multiple hydrogen bond donor and hydrogen bond acceptor sites. And just by looking at this little molecular construct here, you could see how perhaps they'd hydrogen bond and form self-associated complexes. So you could get things like urea-anion dimers, urea-anion tapes, or perhaps urea-urea single anti-stacking. And we actually patented this technology over the last couple of years with our last European patents in 2020, our last US patent in 2020, and we have an international patent filed in 2018 relating to the therapeutic innovation surrounding this class of molecule. So what do they actually do? Well, these very small little molecules are able to selectively interact with different types of phospholipid contained within all of our cell membranes. So when I first started looking into this, I started going, well, could we target bacterial phospholipids over human phospholipids in the cell membrane? We could develop some really nice antimicrobial technologies, right? And then someone said to me that there's not actually that much money in the development of antimicrobials, because when you make a novel antimicrobial, you stick it in a cupboard and allow dust to settle on it because you don't want to use it unless you actually have to, right? So I started thinking, well, what's another way we can develop this technology? So we started to look into developing these things as cancer-enabling cancer therapeutic enabling agents. That's terrible. I'm going to give everyone cancer. This is awful. We're live, aren't we? This is terribly embarrassing. See, I told you I was going to mess up first. Right. But one of the main first changes in cancer cells, right, is actually their change of their surface phospholipid composition. So all of those little polar phospholipids that are normally contained on the inside of your cells, as soon as that cell becomes cancerous, get flipped to the outside of the cell. And this gives us a unique thing to start to target. These polar phospholipids are very similar to the polar phospholipids that we're trying to target in our bacterial technology as well. Okay, so we started looking at these to develop novel anti-cancer technologies, right? And just to prove to you that I can put my money where my mouth is, these are the publications we've got developing this platform technology as functional materials, as broad-spectrum antimicrobials, to selectively interact with phospholipid membranes of different compositions. We've developed a number of novel assays to enable high-throughput analysis of these compounds. We've also been able to increase the efficacy of antibiotic and antiseptic and anti-cancer agents against bacteria and cancer cells. We have shown these things to have potential as drug delivery vehicles. And we have recently shown through a variety of in vivo and in vitro DMPK studies that these compounds do have a druggable profile. We've also put these things into novel flow battery electrolyte technologies. And all of this has been enabled by our reflective practice, utilizing our team of social scientists to help keep the team of researchers that work on these technologies mentally healthy and happy in the workplace environment. So, what kind of groups of people do we work with to enable the development of this technology? Well, we currently have 50 people in international cross-government, industry, and academic consortia. This is termed the SSA Developmental Consortia. And those individuals you're currently seeing on the team, we have Professor Dan Mulvihill. He heads up our microbiology at the University of Kent. We have Dr. Mark Sutton and Charlotte Hind. These head up the antimicrobial approach for what was Public Health England, is now UK HSA. And we have Professor Carolina Paul Albertine, who is a professor of microbiology at the University of the Free State in South Africa. On top of this, we have Jose Ortigo Roldan, and he's helped us with our NMR approach to studying phospholipid small molecule membrane interactions. We have Professor Michelle Garrett. She used to head up a drug development team for Onyx Pharmaceuticals in the States, so if you actually are unfortunate enough to get cancer, you'll end up taking some of the drugs she actually developed. We have Anna McConnell, who heads up our metal coordinations arm of our science. We have Dr. Ewan Clark, who does all of our computational simulation. Dr. Callie Haynes, I should mention, Anna's in Germany. Dr. Callie Haynes at UCL. She heads up our ion transport arm of our studies. We have Professor Hamish Ryder, who is CEO of Cancer Research UK's Therapeutic Discovery Laboratories. We have Mr. Sashi Kumu, who is a consultant cancer specialist up in the University of, the NHS South East Trust. We have the well-known Jeremy Frey, who started off this meeting here, who's convincing me and helping me with all the data that we collect. We have our patient advocacy and social sciences team headed up by Jennifer Lee and Katrina Taylor. And then there's me that tries to figure out what on earth is going on a day-by-day basis. Okay, so what kind of data do we collect, right? What's my email inbox full of? Well, these small molecules, they look pretty simple, but they really are not. So just this small molecular construct in organic polar solvents can form hydrogen-bonded dimers. Stick it in water, and you get the formation of spherical aggregates. Stick in some table salt, heat it up, cool it down very safely with a heat gun, and what you end up with is a change in that supramolecular self-associated structure from sphere to strand, and these things become hydrogels. Okay, so for every single molecule that we produce, just to figure out what it's kind of doing on a very superficial basis, we go through full molecular characterization, including proton, carbon, NMR, IR, melting point, mass spectrometry, and in silico modeling. This is quickly followed up by single crystal X-ray diffraction, quantitative NMR, dynamic light scattering, zeta potential studies, critical micelle concentration determination studies, scanning electron microscopy, transmission electron microscopy, normal, like solution state fluorescence microscopy, and then a whole load of NMR, including variable temperature NMR, proton NMR self-association studies, and DOSY NMR, and all of this data I dumped on Tom. Right. We also develop our own assays to increase the high throughput nature of this technology, right? And in particular, we developed a whole suite of plate reader technologies. This means we can analyze 384 samples simultaneously. And each sample we can get 177 data points out of, which gives us an awful lot of data, right? But then we need to process and store it and figure out what it's telling us, and that really hurts my brain, right? On top of that, we developed a new nanodisc technology. So this is where we take small rafts of phospholipid membrane that we can extract from cells or synthetic vesicles, belt them around with a synthetic polymer, and then we have to do a whole load of characterization via TEM again, and NMR, and some form of UV vis. And then we stick these in an NMR machine with our compounds and run a whole host of titrations. We go through a whole load of integration for all of these NMR spectra that we obtain. And from plotting the relative normalized area of these integrals, we can understand how our molecules are interacting with different phospholipid membranes using a standard solution state NMR machine that any self-respecting university, even the University of Kent now, has with a lovely new sparkly cryoprobe, right? So what other types of data do we collect? Well, we collect a whole load of growth profiles from our microbial growth experiments. So the normal black dotted line there represents the normal growth of, in this case, MRSA. And the other colored lines there represent what happens when we put some of our compound in there and we start killing off this bacteria. But in order to figure out values such as the minimum inhibitory concentration, which is how you basically rank how effective these molecules are as antimicrobial agents, you need to have about 6 to 12 points. So 6 to 12 different graph profiles, which need to be repeated up to 9 times. In addition to that, you then need to do mode of action studies. So this is a mode of action study. We've used fluorescence microscopy to do this, and what you can see is our supramolecular self-associated structures, these green spheres there, bubbling along and interacting, in this case, with MRSA. There's those dark circles there. And then these are just fluorescence microscopy images, and what you see now is only our compound. So our compound is no longer in a sphere form. It's now slapped itself all over the surface of the cell. It's burrowed its way into the cell and is now dive-bombed the nucleoid, meaning there's no return to life for these bacteria. On top of that, I did tell you that these things not only exist as spheres, they exist as strands. So when we get this material formation, we need to do everything from inversion testing to hardcore rheometry to work out stress and strain profiles for these novel materials. We then do a whole load of SEM and TEM and fluorescence microscopy again. And then we need to make sure that these materials still retain their therapeutic properties. Can they still kill bacteria? Not in their spherical state, but now in their gel state. And the answer is yes, they can. But this, again, is more data that's coming into our inboxes in multiple different formats. Moving into for our anti-cancer studies, we do the same similar experiments as we do with microbes, but with cancer cells. But we also have managed to show that these things can enhance the therapeutic activity of things like cisplatin, and that means that every single one of those boxes you see in those charts that I've just thrown up is the result from one of those graphs you see on the left-hand side. That's a huge amount of data that ends up stuck in these to get out, our findings to show that we do have synergy between our SSA technology and the cisplatin. But again, we need to move into mode of action studies after this. We need to figure out how these things are acting. And that means we have to do a whole lot of Western blot experiments, the results of which are shown on the right-hand side of this screen. And again, we have our fluorescence microscopy where we can see our compounds adhering to the surface of this is ovarian cancer cells, permeating those ovarian cancer cells and where those compounds end up. You do all of that and you start celebrating that you might have actually achieved something, and then you go and talk to someone in the pharmaceutical industry that tells you, no, you haven't, please get on and do your DMPK work. So that leaves you with a whole load more data. So what you see here is the output of 3 months' worth of work by the Cancer Research UK's Therapeutic Discovery Laboratories, where they investigated a few of these compounds, figuring out how well they bound to proteins in your blood supply, how stable they were in your blood plasma, how able they were to cross your intestinal membranes. And that's even before you get into mouse model studies. As soon as you get into mouse model studies, you have to figure out at what concentration your agents can get into the mouse's blood supply, get circulated around that body, and then you have to figure out in what concentrations, at what time points, those molecules manage to diffuse into specific mouse tissues, and whether or not you are in a concentration range that enables you to elicit a therapeutic effect. So after you've done all of that, you count up how many years your team members have actually given to you of their lives for you to be able to put out these publications and start writing more grant proposals. And in the last 6 years, I figured out it was 45 years of people's time, and then I felt really guilty. We have had a lot of fun doing it, but this is an awful lot of resource. It's not just money, it's about the length of time spent collecting this data. So we're now at the point where we're trying to get this innovation into the clinic. We have line of sight to the clinic. We have a pipeline and for all intents and purposes, virtual drug company to enable this. But this is our therapeutic test cascade. So these are the experiments we still need to perform in order to get enough data to go towards phase 1 clinical trials, right? So what support do we need in order to enable this? Well, we need some effective data curation because I can't cope with my email inbox as it is, and I certainly can't with all this lot flying at me left, right, and centre. We need high throughput data analysis because trying to raise the funds to employ a PhD student or a postdoc is hard enough. You want to make sure that that time is used wisely and the most effectively it can do. And we also need to identify quantitative structure-activity relationships because if I have to sit down with Excel one more time plotting things on 2 axes, it just doesn't make me happy. Right? And it's at this point that my collaboration with Tom here and the other members of the team who are smirking at the back of the room, I will call you 2 out in a minute, right, started to help me with this and have educated me somewhat in how to achieve my dreams of an organized data system and being able to figure out these structure-activity relationships. So I'm going to pass over to Tom.
Thank you. Yeah. as Jen said, to give a really good introduction into the problem we've got here. So my name is Thomas Allam, and I'm just going to give you a background into sort of the work I've done leading up to this point. My undergraduate dissertation was on modeling and curating of solubility data. I completed a summer internship under this network, which was working with even bigger data sets. So I've had quite a lot of experience handling large data sets and understanding the best way to do that. So I'm currently doing a postgraduate master's research project with Jeremy Frey, looking into the problems that Jen's described with the data curation. So in order to begin any data curation project, it's important to gather the data. So this is really where the hard work in any ML project comes from. So this data, as Jen's mentioned, collaborating with lots of different research groups and people from all over the globe in so many different domains. So this data is siloed and it's not in a centralized location. So the first issue is accessing the data. This data is currently stored in Excel files, and that is just a data point. So you'll have binding constants, but this binding constant was worked out using spectra files. So there's spectra files that exist somewhere where that data point came from, and you've also got text files giving information as to the conditions of those spectras, how they were run. The issue is these aren't very machine interpretable, which is very difficult when you're trying to do modeling on this kind of work. So for example, the diagram in the top right is just a very clustered folder in a format where you don't really know where the data is. You know it's there somewhere, but it's very hard to be accessible. And the problem with these spreadsheets is that these are curated by experts in the field, often to produce a paper. So they're very specified to them and don't actually contain the information required in order for someone else to come in and have a look at it. So you might have a column, as Jen explained, saying MIC, but this MIC may not be consistent across groups, and the methods to get the MIC may vary between the groups as well. So another example of this is in the spectra. So I was given a load of spectra by Jen, and this is just an example of some of the names I came across. Carbon NMR1 folder 11, which is very useful sort of at the time when you're making the paper, but even the same researcher coming back for 4 years from when the spectra was run, you can see where this problem lies. So another problem exists with these data sets is that there's lots of NaN values because this is experimental work, but just to give you an idea of sort of the context needed for running these kind of experiments. This NaN value could exist, so this data point won't exist, and this could be because of lack of solubility, or the experiment was run but it wasn't successful, or hasn't been attempted yet, and all those different NaN values actually mean different things, but are recorded in the same way, and you'd have no idea unless you spoke to the researchers about this. So I've explained the problem, so what's the proposed solution? So initially I came at this obviously from trying to get it into the best format to do AI work for the master's project I'm doing. So the idea was to create a relational database. So as I explained, this data is currently stored in different Word documents and different Excel files. And a relational database allows you to store all those tables, which they are, in a centralized place where everyone will know where it is, and all the data is there. But with the two really key improvements, the data is allowed to be interconnected. So, as Jen was saying, they're looking into the reaction, as an example, the interaction between chemicals and bacteria. And this interaction has a value. So this value is interacted from the chemical side and also from the biological side. And each of these, there's been experiments done on each of the strains and also on the molecules. And all this data can be stored, so computational descriptors as well as the experimental data Jen stored. And it can be there and accessed when needed. Having data centralized allows for collaboration, and this is really necessary for this kind of work because it's so interdisciplinary. Also, it's a way of modernizing it, so there's not a lot that can be done at the moment as all this data is in Excel files. But if it's put in a system like this, there is room for improvement and use of software that already exists to solve problems like this in other industries. So again, it's making this data interpretable to machines and not just humans is the big step we're trying to drive at. And on top of this, because this is currently only allowed to store sort of data points, we can also add links to spectrum storage in order to validate, and also then for researchers to go back and have a look at where their data comes from. So you might be saying, oh, that's all a really good idea, but how has it actually helped? So this is an example of a machine learning model I run. It's called a PCA, a Principal Component Analysis. And as you can see, you might just look at it and go, well, that's just a little graph. But in there is data from 2 different research groups, which was already in different areas of the country, in different Excel spreadsheets. And also the coloring of the molecules was in another spreadsheet, and the MIC values in another spreadsheet. So all this data was needed to put together in order to create this graph. So it's actually the work that goes on behind the scenes. So if the only step needed was to take it from some centralized location and then run these models, that would be really easy. But the actual process I've had to go through is that. So getting the Excel spreadsheets was a process in itself. Then once I've got the Excel spreadsheets, formatting them in a way that they can be put into a database, as well as understanding the context from the researchers. Also, I needed to get over some of the NaN value issues. I had to run some computational descriptors. So this was all a process that's currently done manually. But once the data's in that system, you can create these kind of interactive dashboards, which will allow researchers like Jen, who have all the knowledge about this stuff, to easily sort of drag and drop and create graphs rather than just using Excel, which Jen clearly doesn't like. And the real issue to sort of implementing a system like that is this red arrow, where can we somehow automate all the other things, so by researchers simply just input the data, and that's kind of the crux of the issue here. So some challenges, as you've seen from the slide before, this takes a lot of time, and you don't actually see the sort of lovely machine learning models until a long time into the project, which can be frustrating. It's an interdisciplinary challenge, so lots of domain expertise is needed to actually structure the data correctly in order to put it into a database, and also a large amount of expertise is needed to oversee a sort of data curation project like this. As I mentioned, different researchers have different requirements. So if you're coming at it from a machine learning standpoint, all you want is the data and knowing that data is valid. But from a chemist and biology standpoint, you might want to go back and look at some of the spectra you've already generated, for example. And there's also issues with different approaches within research groups and standardizing these kind of things. So the final slide is some future work. So as I've said, we are laying the groundwork, hopefully, for some really interesting AI to be done on this work. And the next steps involve making the database easily accessible, and then hopefully doing some modeling.
So at this point, I really have to point out that this MRes student who's been working with us for about 5 months, I threw in a meeting in front of those entire 50 researchers, and he managed to gauge the interest of the UK HSA antimicrobial development team who have been supplying him with data as well. So his approach really has changed the way that not only we do research within my group, but also looking towards governmental bodies and having a real impact within the medicinal field. So it only leaves us to then just say thank you to all of the collaborators and the funders so far, and thank you for your attention, and thank you, Tom, for putting on a show in front of a really scary audience.
Well, thank you, Jen. Thank you.